Well, here we are again, and we're going to talk about sex and how great it really is and how dramatic it can change the world. And I should have mentioned before that I'm only wearing this silly hat because I found it in the basement and I thought it would be fun. So I'm doing it. Okay. I hope you like it. Anyway, attached to this uh, unit, I have provided a Word document with these two links. These are two PBS videos that are outstanding. One is what Darwin never knew. What did he never know? He didn't know about meiosis. That's what he didn't know. And the other is life's greatest miracle. Life's greatest miracle is about creating a baby. But in the first chapter of it, and that's what I want you to focus on, there's an animation about crossing over in meiosis that explains why we are all unique individuals. And we're going to talk about that. And also in the first part of this video, a question is asked whether or not we control our desires or whether our DNA tells us what to do, when to do it, and maybe even who to do it with. We know that our bodies send chemical signals called pheromones out into the atmosphere, which makes it to other people, and their DNA picks this up, and their DNA tells them whether they should like us or not. It's an interesting thing, fascinating in fact. So take a look at these two. These are a little over an hour each, and but I think they're well worth paying attention to. They're excellent, excellent videos. So Gregor Mendel, who was Gregor Mendel? Gregor Mendel was a kid who grew up in a well-off family, and they sent him to boarding school. And boarding school in his day was the monastery. And the monastery was where the monks lived and taught. They were the scholars of the day. In fact, the church was the source of an awful lot of scholarly activity. Gregor Mendel became a monk, and he became a scholar. And he did research and kept excellent records. His interest was, how do organisms pass their traits down to successive generations? And next week, next unit, you'll learn about genetics and some of Gregor Mendel's findings and how he did it. But right now, I just want to make the point that Gregor Mendel was on the very verge of discovering meiosis, but he didn't have a microscope to look at the cells and to see the chromosomes and to watch them in living cells. So he was close though, because he developed through his work a law of independent assortment, and we're going to see how that works. We're going to see how that works. And then there was Charles Darwin. He lived from 1809 to 1882, and he, of course, was the first naturalist that suggested why evolution occurred and somewhat how it occurred. And he got done with his trip around the world where he collected all these organisms, especially from the Galapagos, the ones that are made famous, the story of the finches of the different islands. And he published in 1859. He finished his trip and got home in 1831. So it was almost 30 years from the time he started studying the specimens he brought back to the time that he thought he had figured out what was actually going on. And he probably would have even had stronger evidence and had a quicker understanding of what was going on if he had known what meiosis was. Okay, so let's take a look at how meiosis works with our wonderful animation, and it's pretty good one.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells.
Okay, so pay attention. In meiosis 1, the number of chromosomes is halved. Remember that a typical 2N cell, and N is equal to 23 in humans, we have 23 chromosomes. We only have chromosomes during meiosis or mitosis because the rest of the time it's all unwound as chromatin. In fact, the chromatin, which is our DNA, in our typical cell, if it was all pulled out and stretched out, it would be 6 foot long. So it's packed into those cells, but it's floating around in strands so that it can unwind, open up, and send its coded message out into the cell. Well, during meiosis, during prophase, or mitosis during prophase, those chromosomes wind up and take the typical X appearance that we're familiar with, only during that time. During meiosis 1, the number of chromosomes is halved. So instead of 2 times 23 equal 46, we now have only 23. Let's see what happens.
Now let's look at each of these divisions in more detail. Meiosis 1 begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs.
Okay, this is where it all happens. This is where evolution becomes evolution, and where you become you and I become me, all with this one little event called crossing over. And remember, let's just say, for sake of argument, that purple is the chromosome from your mother. There's 23 of these, and there's 23 of these pairs, and the green one is the chromosome from your father. Each of those chromosomes has replicated, so we have a homologous pair. The homologous pair is mom and dad. And this is the part of the video in Life's Greatest Miracle that I wanted you to see, so that you could understand how all of our DNA and our genes get shuffled before sperm or eggs are made. And sperm is made after puberty in boys and before birth. Eggs are made for girls. All the eggs that a girl will ever have are made before they are birthed, while they are a baby in their mother's womb. And it is during menstruation that, one by one, the eggs mature.
During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another.
So you can see we've scrambled the eggs here. We've scrambled the eggs. And this is the part that Darwin never knew. This is what makes every individual born unique, and some more adaptable to the environment than others. Crossing over in Meiosis 1, if Darwin only had known.
In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just as easily have ended on top.
Okay, so now we have scrambled mom and dad. So mostly mom and mostly dad. Mostly dad and mostly mom. But what he's saying is this purple could be on top and the green would be on the bottom. That's called independent assortment, which means that we have 46, no, we have 23 homologous pairs as humans. And the one from mom and the one from dad sort independently for every chromosome. In fact, it's this phenomenon that made the Mendel's work come out so well, was because he knew this was happening. He just couldn't see it. And so he said that the traits sort independently of another. And he was lucky in that his traits that he studied were each on separate chromosomes. If he had been studying traits by accident that were on the same chromosomes, his data would have been so confused he never would have figured this out. So he was lucky. Or maybe the hand of Providence thought that it was time people started figuring these things out. I don't know. But independent assortment, again, is that this homologous pair could be the other way around, and whatever happens to this pair does not necessarily have to happen to this pair, or the next pair, or the next pair, or the next pair. So we don't know how much dad will dominate sperm 1, or how much mom will dominate sperm 1 or sperm 2, but every sperm is going to be uniquely different from each other because of two phenomena. One is crossing over, scrambling the eggs, so to speak, between the individual chromosomes in the homologous pair, and because the different chromosomes sort along that midline during metaphase 1 independently from each other.
Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, two cells are created, and each cell has half the number of chromosomes compared to the original cell.
Now don't let this double structure confuse you. There are 23 of these in this cell for a human, and there are 23 of these in this cell